So, very good morning to all of you. Welcome to the 6th technical session of International Conference on Futuristic Materials. In this technical session, we will have a plenary talk by Professor S. Bhardwaj from Republic of Korea and 3 invited talks by Professor S.K. Dubey from Uttarakhand, Dr. Y. Dwivedi from Haryana, and Dr. Abhimanyu Singh from Prayagraj. So, to chair this session, I would like to invite our head of the department, Professor Shantanu Rastogi, sir. Professor Rastogi, sir, kindly unmute yourself. Thank you, sir. I think we don't have our plenary speaker with us so far. So, should I wait for a few minutes?
Yeah, did you contact him on WhatsApp or mail something? Just to inform him that we are ready.
Let me contact, sir.
Yeah.
Sir, I was informed that he is going to be connected very soon.
Okay. And what about your chairperson?
Sir, you are going to chair this session? Yes, we have Professor Shiv Bhardwaj with us.
So, Ambrish.
Yes, sir.
You can introduce him, then I will.
Okay, sir. So we have our plenary speaker, Professor S. Bhardwaj with us. Let me also welcome co-chairperson of this session, Professor Umesh Yadav, sir, from Department of Physics. Professor Umesh Yadav, sir, please unmute yourself.
Okay, thank you.
So I would request Professor Umesh Yadav, sir, to introduce our plenary speaker, Professor Shiv Bhardwaj.
Has you joined?
Yes, sir, he has joined. Okay. Professor Bhardwaj, please confirm you are with us.
Hello.
Okay.
Hello.
Yes, sir, we can hear you.
Good morning to all of you. Yes, sir, I can hear you.
Okay, very good morning,
okay, sir, thank
you all. So this is the technical session number 6 of this international conference. In this session, one plenary talk will be here by Shiv Bhardwaj, and there will be 3 invited talks. So the first plenary talk in this session is by Dr. Shiv Bhardwaj. I know him since 2018, and we are working on collaborative projects with aimed applications of nanotechnology and drug discovery. While working and interacting with him, I realized that he is established researchers in the field of nanotechnology, drug discovery, medical chemistry, biochemistry, and biotechnology. He has excellent experience and professional expertise in several experimental methods like animal cell culture, genetic engineering, protein chemistry, nanotechnology, and various biophysical techniques. Also, I have learned that he has developed proficiency in computational and CADD techniques for intense molecular modeling, pharmacophore modeling, docking, and molecular dynamic simulation, as evident as from published articles in internationally reputed high-impact journals. Besides, he has outstanding communication skills blend with professional scientific writing and project management makes him an ideal candidate for both R&D and management positions. I believe Dr. Bharadwaj is a highly motivated, dedicated, smart researcher with sincere social etiquette. He has a warm and cooperative attitude towards colleagues and companions and an excellent team player for individual and teamwork. With these few words, I invite Dr. Bharadwaj to present his talk on advanced nanomaterials for medical applications.
Uh,
Please.
yes. Thank you, sir. Thank you so much for wonderful introduction. Uh, so let's begin with our talk. Can I share my slides?
Yes, sir. Yes, sir.
Uh, I think maybe you need to make me as a presenter, assign me as a presenter so I can share my screen. I cannot actually. Hello.
You have already the permission to share your screen. Please go ahead.
Okay.
You are co-host, so you can.
Thank you. Okay. So is it visible to everyone?
Yes, sir.
Yes.
Yes.
Okay, thank you so much. First of all, thank you again for inviting me for this conference. So without taking so much time, I will continue with my talk. So topic of my talk is advanced nanomaterials for medical applications. So in this particular talk, I'm going to talk about a very briefly about the nanotechnology, what is nanotechnology, and about the dimension of the nanotechnology. Then I will give a brief account on the history and the type of nanomaterials, and then how these nanomaterials are advantageous for medical applications. And then I will give a little bit discussion about advanced nanomaterials and their types, and then their application in medicine. So now, since we all of know that what is nanotechnology, but I would like to give a little account here. So nanotechnology is the study of controlling the matter at a atomic scale, or we can call it molecular scale. So generally, nanotechnology deals with the structure size between 1 to 100 nanometers, but up to 500 nanometer, we may call it, nanomaterials. So but generally, the very sophisticated definition is to consider the materials from 1 to 100 nanometer under nanotechnology or under nanomaterials. And then this nanotechnology involves developing or modifying these nanomaterials or devices within that particular nano range. So to have to have a clear perspective, we can assume that the diameter of an atom range from 0.1 to 0.5 nanometer. And if we talk about a paper sheet, it's about 100,000 nanometer thick. And if we talk about a marble, a nanometer, then 1 meter would be the size of our planet Earth. So how small is nano small? So here an example with respect to different molecules, and then with respect to materials and biological molecules. So for example, if we look at the water molecule, the range is from 10 power minus 1 to, and then if we look at the glucose molecule, it's only 1 nanometer. And then we talk about biological antibodies, we have a size of 10 nanometer. And if we talk about virus now, everyone knows about the COVID-19 and then SARS-CoV-2. So it has a size of about 10 power 2 nanometer. And then bacteria comes in the range of on average 10 power 3. And then we have cancer cells between 10 power 4 to 5. And then if we put a period or a full stop on the paper, that would be a small size of 10 power 6. And then if we talk about tennis ball, that range from 10 to power 8. So these are some examples of materials around us in nanoscale. So down in the slides, you can see this nanomaterial. So these nanomaterials actually fall in the range of 1 nanometer to 10 nanometer. Examples are like liposomes, and then quantum dots, or gold nanoshells and fluorines. So normally the nanomaterials we in our nanotechnology, in nanomedicine, we consider them between 1 to 100 nanometer. So here is some interesting fact to make it more interesting to learn about the nanosmall. So for example, you can see in the image that this is the single human here. And then on the back side, you can see the nanofiber. So a human ear is about 100,000 microbyte, but the nanofibers, they are size of from between 1 to 100 nanometer. So for example, if we hold a hair in our hand, and if your body size reduced to the point where the size of the hair was just 1 nanometer thick, you would be able, you would be like as small as the thickness of the ear you are holding in your hand. So you can imagine how small nano we are talking about, or how small the nano scale is. So if we look at the history, so the concept and idea started on December 1959 with a talk entitled "There's plenty of room at the bottom" by physicist Richard Feynman at an American Physical Society meeting at the California Institute of Technology. Feynman described a process in which the scientists would be able to manipulate and control individual atoms and molecules. That means the nanotechnology is not but the manipulation of the atoms and the molecules. Then over a decade in his exploration of ultra-precision machining, Professor Tunguchi coined the term nanotechnology. But the modern nanotechnology begins only in 1981 with the development of scanning tunneling microscope that could see individual atoms. But MIT researcher like Dexter popularized the term, normally the modern term, this nanotechnology we are using, with his publications Engine of the Corrections in 1986. So we have now different types of nanomaterials. So as we know that the nanomaterials are in the size of in the range of nanoscale, that is from 1 to 100 nanometer. So based on the composition or based on their physical characteristics, we make classified nanomaterials into major 3 types. The one is nanoparticles, second is nanoclay, and then third is nanoemulsions. So I'm not going to talk about all the nanomaterials in this particular talk, but I will briefly describe about some, like under the nanoparticles and then advanced nanoparticles. But this is a general overview about the different types of nanomaterials. So for example, if we look under the nanomaterials, we have again 2 types of nanomaterials. We have inorganic nanomaterials and then organic nanomaterials or nanoparticles. For example, in organic one, we have mesosporic silica or silica nanoparticles, we have gold nanoparticles, we have zinc nanoparticles or other metallic nanoparticles, even cesium oxide nanoparticles or tungsten like that nanoparticles. Then we have these supramagnetic nanoparticles that, of course, these are iron oxide nanoparticles, but with a magnetic nanoproperty. Then we have quantum dots, and the quantum dots are special nanoparticles because they are fluorescent in their nature. So normally they are in the size of 5 nanometer, and then they give the fluorescence, and that's why we call them quantum dots. And then under the organic nanomaterials, we have liposomes and then these polymeric nanoparticles like measles and endomers and all. And then under the nanoclay, if we look into, then we have these polypropylene nanoclay systems and all. And then other side, we have these nanoemulsions, which are actually kind of mixture of oil and water. And then based on that one, we can make water into oil or oil into water nanoemulsions. And based on the characteristics, we are normally use the different nanoparticles in different application. If I talk about the nano emulsions, so normally these nano emulsions we use in the medicines because they can easily entrap and pack the drug molecules. And then there are many already medical products are like skin creams available which contain these nano emulsions. And similarly, if I talk about the super paramagnetic nanoparticles, these super nanoparticles and the inorganic nanomaterials normally used in brain scanning or MRI scanning because they have some magnetic effect and then can easily be uh, uh, do the image contrasting and all. So similarly, if I talk about the quantum dots, so the quantum dots, since they are fluorescent, so normally in not much had been done in medical science in the body, but outside the body, these nano particles are being used for diagnosis purpose. For example, as a detection kit for the disease or some biomarker. So similarly, based on the application or based on the need, these nanoparticles can be modified. For example, mesosporic silicon nanoparticles can be functionalized with some other particles or some compound, and then according to requirement, we can use them. But I will talk about a short example or certain example with the already published articles, and then I will give a brief introduction or like brief overview on my research, what I have done with nanoparticles, and then the research being going on in our group. So now can talk about the advanced nanomaterials because we understand what is nanomaterials. So advanced nanomaterials are again nanomaterials, but they are being called advanced nanomaterials because typically they have a greater physical, electronic, and optical properties compared to the other nanomaterials. For example, there are many advanced nanomaterials being developed and being published. So I have given you some examples of some of the advanced nanomaterials. For example, we have block polymer nanofibers or nanowires and nanotubes. Now we know what is nanofiber, nanowires or nanotubes, but co-block means like copolymer means that we are using two polymers together and then doing their polymerizations and then forming some these nano matter. So for our application, for example, these copolymer nanofibers or nanowire or nanotubes kind of nanomaterials belongs in a range of 100 nanometers and then may go up to like meter of or micrometers of length. And mostly these kind of polymers or these kind of nanomaterials are used in tissue engineering for growing the cell culture, or maybe in if we have suitable polymer, then we can use them in 3D or 2D tissue culture also. Then we have a second type of advanced nanomaterials. These are, we call them smart nanoassembly. Again, because these are smart these are smart nanoassembly because these nanoassembly means that we have some code units, they come together and then form the assemblies, uh, because we are not doing any external efforts, or not we are putting some external energy into it, because the process is its own. And so the term is smart for these nanomaterials. And these nanomaterials again fall in nanoscale, that is less than 100 nanometer. And then according to our requirement and according to the co-polymerizing agent, or we call them molecules, or these what you call the molecule we are using to form the assembly. So according to that, we can pack our drugs, or we can also make some kind of detection with these nano assemblies. One popular example is that stimulus-based drug delivery. For example, now we know that in the cancer, there is acidic pH and there are certain lipases also. So smartness and nano assembly, we can make some nanoparticles which are mm, pH sensitive. That means the polymers comes together, make the composite, and which are sensitive to the pH, that is acidic pH in the tumor. So these nanoparticles will form, and then when they go to the site, they will dissolve there, and then they will release the drug. So like this, we have these smart nano assemblies, and then some companies or industries are already doing some research for drug delivery. And then we have another type of advanced nanomaterials. Among them, we have helical mesoscopic structures. So helical mesoscopic structure means these are the nanomaterials with helical structures. Example is nucleic acid derivatives or nucleic acids. These nucleic acids are used to form some super helical structures, like maybe they form tubes and discs, and then they again use form for different function and according to the property. For example, for the detection outside the body, or maybe for drug delivery in the body. So uh, these are some advanced nanomaterials. And next we come to metallic nanoparticles, or we have nanotubes or nanourchins also. So as the name says that metallic means that we are using metals here for formation of nanoparticles. An example is gold, zinc oxide, cadmium oxide, aluminum oxide, titanium oxide, and so on. So based on the structure, these nanoparticles could be spherical, so we normally call them nanoparticles, and then they could be tubes, that means they are hollow inside, and then they may look like sea animal urchins, so we call them nanourchins. So these nanoparticles normally they are used outside the body, not in the body. Like I said about the iron oxide before, under the different types of nanoparticles. For example, zinc oxide and gold, we can make them composite, and then we can use them for diagnosis purpose. For example, uh, gold is actually least reactive element, so we can make some kind of paper strips and biosensor strips already available with the gold, and then these strips are being used for the detection of some cancer biomarkers, and then some antibodies, or some people are if we can just functionalize this nanoparticles with the thiol group, that is very easy to functionalize the nanoparticles unlike other nanoparticles. So we can easily conjugate some antibodies on it, and then we can use this nanoparticle even for the detection of some like I said, antigens and all. So that means normally these nanoparticles are being used in sensing. So another application is that these nanoparticles, for example, gold and silver and zinc, they have highly scattering efficiency. So these nanoparticles are used in the substrate formation for SCR sensing, that is a Raman scattering. So there's another type of sensing. So in short, that these kind of nanoparticles are normally used in medicine, or by some companies in the development some medicinal or pharmaceutical products for the diagnostic purpose. Next, we have carbon nanomaterials, and then or we carbon nanoparticles or nanotubes. So basically, carbon, as the name says, the carbon nanoparticles. So these particles or nanotubes are made up of totally 100% carbon. An example is graphene, and then carbon nanotubes. So what happens where we can use these nanoparticles? So these nanoparticles, for example, these carbon particles, for example, graphene and CNTs are being used in tissue engineering. Basically, they are using these nanoparticles for growing the cultures. For example, these neural cells, because they are not easy, we cannot easily grow them in lab, because we do not have that physiological or biological this environment available outside the lab, body. So these materials are being used as a scaffold formation or designing, and along with some other polymers, or maybe alone also. For example, graphene oxide or reduced graphene oxide being used in culturing, and then they are checking how these nanoparticles can be used for the culturing purpose. Another application is that these nanoparticles are highly antimicrobial function. So these some materials, these medical instruments are being targeted with this nanomaterials coating. So for example, pseudomonas normally form biofilm in medical apparatus. So these nanoparticles are being targeted as a coating agent on such these tools used in medicine, or in medical applications, or in hospitals and all. So We have another type is this hybrid or composite nanomaterials. So as name says that these are hybrid. In this among this type of nanomaterials, we have a composition of we are using carbon nanomaterials along with some polymers or in the very first one I said that the nanofiber or nanowire, polymeric nanofiber, we are making composite of them, and then mostly these are being used for tissue engineering again, because they provide more sophisticated biological environment, because the polymer we are using, it's a biocompatible, and then we can easily design them, mold them to 3D scaffolds in tissue engineering or regeneration medicines. Another application is that these nanomaterials, hybrid nanomaterials, are used for mimicking the bone structures, so that they can easily help in the bone healing for the bones and all, or fracture. So something like that applications are being exploited, are being considered for these hybrid composite nanomaterials. So now we're talking about so much nanoparticles. So why this? What makes technology at the nanoscale different from the technology at the macro scale? So this is, of course, a question arises in everyone's minds, that why we are talking about nanomaterials, and when we can see the things with our naked eye, and then the nanoparticles, which is hard to see with the naked eye. So why we should talk about the nanomaterials and not use these macroscopic particles? So the major reason behind that, the properties of the particles of the material at nanoscales get changed. What are those properties? The properties like melting point, fluorescence, electrical conductivity, and chemical reactivity. So when I said about this graphene nanoparticles, so I said in an example that these graphene nanoparticles are being used in the culturing of neural cells. So as we know, the neural cells need conductivity, or these neural signals, electrical signals, and in the transportation to communicate with each other. So that's why we need a material which is conductive in nature, and then assist in the conduction of this electrical signal. So here we come with the graphene, and in present today research, we know that the graphene is highly conductive in nature. So now we understand that graphene, because graphene is at a nanoscale, is almost one single layer of atom, is very highly conductive, and it do not have so much resistance. So that's why the nanoparticles have these specific properties. Then we have this chemical reactivity. So what happens then, when the surface is larger, the greater amount of the material comes into contact with the surrounding material and increases the reactivity. So that means surface to the when the surface when we reduce the particles, the surface area increases. So these nanoparticles have high catalytic reactivity. For example, not in the medical purpose so far, but some research being conducted, and I will give an example here. For example, the nanoparticles are being used in industry for hydrogen production as a fuel. So that means that we can use nanoparticle as a catalyst also. And then here come with another thing is the tunable property. Tunable property means the nanomaterials can be tuned by varying the size of the particle. So for example, the changing the fluorescence color, so the particle can be identified. So like I said about the quantum dots, so the quantum dots, smaller the quantum dots, so they have like a bluish or yellowish color, but when the particles size increases, so they give a fluorescence in the red zone, or red fluorescence. So based on these different nanoparticles, we can do bioimaging of different body parts. For example, in bioimaging, we can use one type of, for example, green nanoparticle for only for muscles staining, and then other one we can like red one, which are for the neural cells, which are deep down in the tissues, so the tissue do not absorb red light so much. So we can use these nanoparticles with the red fluorescence for staining the neurons or nerve cells. So that means that when we are considering the nanoparticles at the nanoscale, so we can easily tune them, and then according to our requirement, we can we can achieve the our these desired results. So another thing comes with the small size or nano size is that the complexity. So what happens here then? The complexity offer a variety of functions to the product. For example, we have a carbon derivatives or these fluorines and the carbon nanotubes. They show a negative curvature. So the negative curvature, because of that, uh, these nanoparticles possess some unexpected electronic and mechanical properties. That means these materials are very strong, and then they have high conductivity. So based on we can use or we can exploit this high conductivity and then their mechanical strength in a desired application. So where what are the different industries where nanoparticles are being or nanotechnology are being used apart from medicine? For example, in automatics, I mentioned before that nanoparticles are being used in the fuels cells, these fuel cells and filters, bending protections, nanoproducts, and for example, in chemical industries also they are using nanotubes, nanocomposites, and then cosmetic creams. For example, nanoparticles are being used in UV protection. For example, in cosmetics, these nanoparticles, for example, zinc oxide, are being used in cosmetics. So they are known to this protect the skin or epithelial skin from the UV light, like titanium oxide. So people normally they are using a composite of zinc oxide and titanium oxide in the this anti-UV protection. So another application is their environment. For example, they are using for biodegradation, filtration, and then removing of impurities, marking of food, and desterizations. For example, filtration, the graphene can be used, and then it can be easily used to filter the seawater. So because the graphene has the ability to absorb the metal, or we can say the salt on its surface, and then let the water go through it. Another example is that food marking and all. So we can use this antimicrobial ability of nanoparticles in food coating, so we can protect our food from the these food pathogens and all. Another is electronic. For example, I talk about the graphene. So graphene is highly conductive. So this graphene is being tested in quantum computer applications and all. So similarly, we can use this graphene in biosensor device designing and all. So another application is military. So in the military, like people are using the graphene for like respirators, fabrics, providing biological or chemical protection, hemostasis pads and all. And then even it's being used for example, graphene in the coating of this what you call this first aids, where they can easily use it to protect the damaged tissue from some kind of viral or some microbial infection or fungal infection, because like I mentioned before, this graphene is antimicrobial properties. Then we have textile industry. In the textile industry, the people are using in, like I mentioned here, the sport clothing, novel apparels, hydrophobic and non-foiling fabrics. For example, some nanoparticles are repel the water, so people are using this kind of nanotechnology to have these waterproof clothes and all. So next we come with the medical industry. The medical industry, the nanoparticles are being used in either for the drug delivery or making artificial joints, and the tissue replacement or tissue engineering. So these nanoparticles may be pure form or in a composite form being used in medical industry. So in this talk, I will give a little not so much explanation, but some I will explain some examples. I will discuss some research, the application of these nanoparticles in medicine. So, like I said, the application of medical nanotechnology, uh, it has crossed many areas in this field. For example, nanotechnology is being used in diagnostic of disease, abnormal conditions. Diagnostic of disease, for example, now everyone these days, like in India, I would say like 10 I mean, out of 10, 5 or 6 people are suffer from diabetes. So now everyone would have seen this device, biosensor device, which they put the blood drop and then they easily measure the concentration of sugar in the blood sample. So the device itself, the chip itself, is contain graphene. The advanced chip coming in the market, they contain graphene, and they are more sensitive compared to the metallic one, the other one. So these chip containing the graphene or graphene oxide or some polymer be the graphene for this chip uh, are very sensitive to the glucose, and then help in the estimation of accurate estimation of the glucose level. So that means the diagnostic one. Similarly, we can use this nanomaterials for like some cancer markers or similar like that, or maybe some hormonal disimbalance, and also such abnormal or disorder conditions can be predicted using the nanotechnology devices. Then next come in the drug delivery, therapeutics, gene therapy, and gene silencing. So what happens in drug delivery normally, for giving an example of a chemotherapy. So in the chemotherapy, if someone has a tumor deep down in the tissue, for example, in the spleen, so this person is being treated using the chemotherapy. They just inject the drug, and then the drug flow through complete body, whole body, and then give its effect on the tumor. But this using nanoparticles, we as earlier discussed, we can do the site directed drug delivery. So that means the nanoparticles will go to the site of the tumor and then will open there only, and then will deliver the drug, so that we can minimize the side effect of the drugs. So another example is gene therapy, so, and gene silencing. These are the new approaches of nanoparticles being used in medicine. For example, we have heard about the CRISPR and all. So this is gene therapy. Similarly, this technology CRISPR can be used for the gene silencing, also where we are knocking out the genes, and then expressions of the particular gene will be down. So another example is tissue engineering. I have already spoke about it. The tissue engineering, we are growing the neural cells and all. So we can easily replace the damaged tissue. For example, in case of Parkinson and our Alzheimer, so research is being done in Japan, where they are using this advanced 3D scaffolds of nanomaterials, and then they are placing it within the patient with the stem cells. What they are doing, actually, let me give a brief overview on it. They are collecting the cells from the patients, and then converting them into induced pluripotent cells, and then converting them to neural cells from there. And then these neural cells are being grown on these nano engineered scaffolds, and then when they have a particular confluence, then they are placing them back into the Parkinson or Alzheimer patients. So where this tissue, I mean these nanomaterials or this scaffold is being degraded by the cells, and then the cells replace the damaged cells in the body. So this particular approach is being under clinical trials in Japan. So that's another application of nanomaterials I just told. And then we have in the general sake of increasing the knowledge of the human body, so these nanomaterials are being tested on the human body or some other biological applications to test as how they can affect the body. Uh, uh, for example, we have these silver nanoparticles and gold nanoparticles. Some people says that they have like immunodepletatory effect. So really they do have or not. Uh, I mean, scientists are working in that particular direction also. So I was talking about the sensing and all. So in these disease diagnosing, here's some example. For example, how we can do the biosensing using the nanomaterial. So uh, in this particular figure, you can see that we have three examples. One is FRET, and then other one is electrochemical one, and third one is electrochemical. I will discuss all three briefly. The first is FRET. So FRET is fluorescence energy transfer, Fourier resonance energy transfer. What happening here? The nanoparticles are targeted or maybe conjugated with some of this fluorescent agent. So they are being placed at such a distance that from the other nanoparticle, so which is again fluorescence. For example, here we have this composite of silver and gold, conjugated with some proteins and then conjugated with the nanoparticles. So when some for example, when now they are in a close contact with each other, so the energy from this particular molecule, the fret molecule, conjugated with nanoparticle by thiol group, or this thiol bond, can easily transfer the energy to these nanoparticles, and then the nanoparticles give a particular signal corresponding to the absorbed light. But under antigen or analyte, this light can be blocked. So how much light is blocked, corresponding to that we can have the reading, and then we can measure the amount or concentration of our analyte. Similarly, here in electrochemical one, we have this modified CNT carbon nanotubes with the nanoparticles, gold nanoparticles, and again conjugated with the nanoparticle with the optimer. This is haplomer actually here, DNA sequence. So what happens here when, and then when the when we have the analyte, and when analyte bind with this particle here, so there is a change in the redox potential, and corresponding to that we can calculate the concentration of our analyte in the sample. Similarly, here the analyte can be detected by some antibodies, and then this analyte is conjugated with the primary antibody, and the another secondary antibody is conjugated on the nanoparticle. So the same principle on detection, there's a change in the electrical signal, so the analyte can be detected. This is the normally what we use in this diabetes, like I said, this uh, glucose or diabetic device. We are putting our blood sample, and then we are checking the signal. So that biosensor device is based on electrochemical one here. That this is how the process happen. Another one we have is photoelectrochemical one. Here what we are using, again we are using this nanoparticle, that is quantum dot here. For example, this, but the difference is that when the light fall on the substrate, so there is a flow of electron induced by the photoelectric fact. So corresponding to that, we again check the change in the this current flow, and then that current flow, change in current flow corresponding to the amount of the analyte. So that is how we can again do the sensing of this analyte. So this analyte can be a antigen, or like it could be pathogen like bacteria, or maybe virus, or it could be some biomarkers like mRNA or some other biomolecule in the body. So one example here, a very simple one to understand how this sensing works here. So this is the paper published by Jiu in 2013. They use functionalized gold nanoparticles for the colorimetric detection of dopamine. So let me tell you, in sensing we have two three types of sensing. One is optical sensing, or another one is SCR sensing, and then we have electrochemical sensing. So this one is optical, and optical, under optical we have colorimetric sensing. So in the colorimetric sensing, or in this paper, what they have done, they just take the gold nanoparticles and then conjugate it with the specific optomers, which optomers are specific to the dopamine. So when the nanoparticles with the dopamine are free, that means there is no dopamine in the solution. So these nanoparticles do not conjugate or do not aggregate. So in presence of dopamine, they come together and conjugate. So you can see here that with the increase in concentration, the solution color change. So from light pink to this dark blue color. So corresponding to that, we can detect the amount of the dopamine in this in the our sample. For example, in this study, they detected the dopamine in the human urine, and then human serum sample also, with the amount of 0.16 micromolar in the urine, and then this serum sample, the minimum this detection was 0.163 again. So almost same detection level they in the urine, and then the serum sample they detected using this simple colorimetric detection method. Another one application in medicine is that drug delivery. So under the drug delivery, we can use different type of nanoparticles. For example, we have carbon nanotubes, metal nanoparticles, dendrimers, polymer particles, hydrogels also. Hydrogels are being used for bone healing also, and in tissue engineering also. And then we have this popular one these days, protein drug conjugates, because this is kind of drugs. This is kind of side directed drug delivery. Then we have liposomes and solid phase hybrid particles. These all different particles are being used in drug delivery. So how we can use these nanoparticles in drug delivery? For example, the size varies from 100 to 1 nanometer, and then we can modify the surface. That means the nanoparticles are tunable. So how we can modify the surface? We can do the pegylation or other coating, or we can do some functionalization, surface functionalization. For example, thiol or amine or carboxyl group can be easily put on the surface of a nanoparticle. Then by electrostatic interaction or surface charge, we can also modify the surface, or we can load up a drug molecule. Then we can also do some target ligands. For example, we have antibody, peptide, and aptamers and all. So and another thing is that we can use the different shapes of the nanoparticles. So different shape means that if we are changing the shape, so according to requirement, we can have a desired property. For example, if we are using the sapphire one, so normally sapphire one have this more surface area. So similarly, like this, we can use this nanoparticles for the drug delivery. So one example under this drug delivery
Dr.
is
Bhardwaj,
yes, sir.
you have just 3-4 minutes. Please try to conclude in 3-4 minutes.
Okay, yes, thank you, sir. So another example is here that the silicon nanoparticles, where they use silicon nanoparticles conjugated with the molecules, and then this molecule helps across the blood-brain barrier, and then it helps in the reduction of IL-17, and then this helps in the recovery of spine cord injury. So this was another article published. So next one, we have Thronistic Agents. These agents, actually what they do, we can actually simultaneously monitor the disease, we can do the therapy, and then we can diagnosis also. And among them, for example, here this particular study, they designed this kind of Thronistic using the DNA scaffolds, where they conjugate the nanoparticles, and then using some antibiotics. So this easily detect the bacteria, and then they kill the bacteria also. So another one is the very popular these days is Disco, and this particular technology, they can actually do the whole animal or whole body imaging. For example, this technology, we can easily see the nerves, and then we can see the muscles and other organs also. They are using this nano-based amino labeling. So my contribution, actually, I use this kind of graphene nanomesh hybrid nanoparticles for detection of these pollutants. You can check this one online easily. So running short of time, then my another contribution using the computational one, I figure out that cesium nanoparticles are good for the inhibition of this alpha cycline, which is one of the target in Parkinson. And then I am continuing my research using the graphene composite. For example, this is a graphene foam I designed for the tissue engineering. And then I have designed some other nano composites also. For example, the silver zinc graphene nanofiber composite for CR sensing, similarly for nanospec composites also. And this is another interesting research being going on by GlaxoSmithKline. They are working so-called tiny sensors, which they also call electroceuticals. This is a future medical technology, nanomaterials technology, where they, this using these tiny sensors, we can detect the disease within the body, and then we can destroy the disease within the body, for example, tumor. So to conclude, I will use this quotation from this Semble 1996 Nobel laureate. He said the human health has been always been determined on the nanometer scale. This is where the structure and the properties of the machines and the life in every one of the cells in every living thing, the practical impact of nanoscience and the health wealth will be huge. So with this, I conclude my talk. Thank you so much.
Yeah, thank you, Dr. Bhardwaj, for this interesting talk about use of graphene and other nanomaterials. We will have a discussion through chat box, and probably we'll be circulating questions and you can answer them. I'm happy to inform that we have with us Professor Ajay Singh, who is head of the Department of Zoology in our university, and he is also Dean of Student Welfare. He has been kind enough to chair the session from, so I want to hand it over to him. Professor Ajay Singh. Yeah. You can unmute and start. Yes.
Thank you for you giving me a opportunity to chair the session. Thank you very much, the organizers, and also thankful to Professor S. Bhardwaj. He gave a nice lecture on the application of medical nanotechnology in diagnosis of diseases, drug delivery, gene therapy, gene silencing, and the tissue engineering. Thank you, sir, for your nice lecture on this topic, and I hope this lecture is more beneficial to our students and faculty members. Thank you very much, sir, for your nice lecture. Thank you, sir. And I will invite the second invited lecture given by Dr. S.K. Dubey, University of Petroleum and Energy, Dehradun, and their topic is Understanding Materials Under Irradiation via Modeling and Experimentations. I will invite Professor Dr. S.K. Dubey ji for your lecture, sir.
Thank you very much, sir. Yeah, I've been the student of Gorakhpur University. I did my bachelor's and master's from this department.
Most welcome, sir.
Professor Santanu Rastogi, sir, head of the department. Professor Sugrinath Tiwari was my teacher. He taught us thermodynamics when I was a student in Gorakhpur University. And Professor Umesh Yadav, he's my friend, my colleague, classmate, and he's a professor in physics in Department of Physics at Gorakhpur University. Professor Ajay Singh, chair, sir. So thank you very much for inviting me to this talk, to this virtual conference on the materials. And I would be I'm really thankful for the department and Professor Tiwari, Sugrinath Tiwari, sir, to give me this opportunity for presenting my passion, my area of research in material modeling. And after coming to India from States, I've been working in experimentation also. So I'll share my slides and then I'll start my talk. So can I share right now? Share screen.
Yes, sir. Yes,
Please
sir.
share it, sir.
So is it visible? Are the slides visible? All right.
Your slide is visible, sir.
All right. So this is what you see in the title is my passion to understand materials under irradiation. Basically, this is a part of a broad topic, broad area of research, the materials under extreme conditions. So I am Santosh Dubey. I am currently heading the Department of Physics at UPS, and which is called University of Petroleum and Energy Studies. And for the modeling work, I collaborate with my advisor, Professor Antar Al-Azab from Purdue University, USA. So, and when we say materials under irradiation, so our mind goes to the materials or the components used in nuclear reactors. So what happens in nuclear reactors? We have core. I'm not talking about everything about the nuclear reactors. The thing where the real physics happens. So at the core of the nuclear reactors, you have these nuclear reactions going on. Uranium breaks into fission fragments, you get neutrons, and you get all kind of radiation and so on. So, but what is the flip side of it? Like after the nuclear reactions happen, fission fragments, high energy fission fragments, and neutrons, and all those radiations, they come out, and then they strike, they interact with the surrounding walls, the vessel wall. So what will happen with the vessel wall? So these high energy particles, they impart, or they dump their energy to the vessel wall. So when they dump their energy to the vessel wall, what happens exactly at the microscopic level, at the atomic level? The energy of these particles which are coming out from the nuclear reactions, this is transferred to the atoms in the vessel wall. So when the atom which is released because of the energy transfer from the energetic particles, which is due to the nuclear reactions, the atom which gets released from the lattice side, the first atom which gets released, called primary knock-on atom. And this primary knock-on atom is highly energetic, and it goes inside the lattice, and it leads to secondary, tertiary, and all kind of displacements of the atom. So all of a sudden, lot many atoms, near the interaction region, all these atoms get displaced from the lattice sides. So we have a displacement cascade, formation of a displacement cascade. Now the structure and the property of displacement cascade depends on the energy of the radiation, energy of the primary knock-on atom. So if you try to see the structure of the disturbed region, or you can say the damaged region, so mostly at the center of that damaged region, you have kind of vacant spot, lots of vacant spots, and those lots of vacant spots, you can call them as vacancies. And the atoms which are released from these vacant spots, they can they go and sit in either at interstitial spots in various configurations, or they make split configuration of interstitials. That's also called dumbbell interstitials. So the structure of this damaged region has a very peculiar kind of structure. So at the center you have mostly vacancies, and at the rim, you can say at the outside, at the periphery, you have interstitials. So this event, or this interaction event, which leads to formation of residual concentration of defects, interstitials and vacancies, and also their clusters, this is called radiation damage. This is the fundamental process which happens when the energetic particle, interacts with the material. So keeping this in mind, so think about the fate of a material which is being used inside nuclear reactor for, say, 10 years, 15 years, and so on. So we'll have a multitude. We'll have non-equilibrium concentration of such defects. So we have these kind of damaged region all over the material. So the fate of the material, what will happen to the material after all these interactions with the energetic particle? The material will be highly damaged, at the macroscopic level. Highly damaged, I mean, like you have lots of defects, vacancies, interstitials, and you have the clusters of these defects. All right, so this has far-reaching consequences. I mean, if suppose you have a material, a pure material has very less amount of less defect concentrations. But if you have highly damaged material, so if you have highly damaged material, all kind of phase transformation can be possible. So there are certain observations that I'm showing here. Like you see a stainless steel rod on the left-hand side, which was placed in the nuclear reactor in an unirradiated rod. I mean, before irradiation. After irradiation, after several years, the physical dimension of this rod has increased to 20-30%. This is called a swelling process. So when think about in a situation in which you are using components in nuclear reactor setting, in which you are using a dimension based on various measurements. Now all of a sudden, after a few years, the physical dimension of those components increased. What will happen to the integrity of the material? Inside, everything will be under strain and so on. So this is a very important thing that we need to understand why exactly is happening. Similarly, the creep deformation. Creep deformation is also a very important thing, especially at high temperature. Creep deformation is very, very prominent and very, this is an unwarranted thing. So, and especially under radiation, because we have a lot of, we have non-equilibrium concentration of defect. Creep, the rate of creep is, becomes really high. You can also see a loss of ductility in the materials. The material used before irradiation, it was highly ductile, it has good ductility property. But after irradiation, the material loss ductility, and also have irradiation hardening issue. Like you begin with a material which was really nice, a good deformation region, but as you use that material under this irradiation, hardening increases. I mean, you have the material which you use, it has good elastic properties, but as time progresses, the material becomes hard. You also have compositional instability. Like you started with, for example, say inward alloy, which is used in structures, making structures, and all of a sudden you see like the phase separation kind of things. Spinodal type instabilities are also observed under irradiation. So, and in such cases, even though you anneal, because using annealing process, most of the inhomogeneities and compositions can be taken care of. But if there is some spinodal type, instability, which can't be reversed back, even though you do annealing. So all these processes are really, damaging to the material, and the purpose for which they were used. These observations tell that, because of irradiation, such purposes can't be solved. So at the end, we need to understand what exactly is happening in the material under irradiation. You can also have segregation and precipitation. Segregation means like you make an alloy for certain properties, like for example, stainless steel. It is made stronger by adding carbon to it. It is made non-magnetic by adding chromium and so on, chromium and other things. And it is making so if something happens, so the whatever alloying components you are adding in tracer amount, suppose those alloying components, they go somewhere else. They go to say defect sink, for example, near the at the grain boundary or near the free surfaces and so on. So the property of that material will be different. So segregation is one more process which can lead to loss of certain material properties. And if the segregation happens to a significant amount, so that above the solubility limit of that component in the matrix, so you'll have formation of the precipitates and so on. So what do we need? I mean, at the end of by seeing all these observations, these observations are real. I mean, these have been observed in the nuclear reactors, and that's why they have been reported. So now to understand what exactly is happening, suppose you want to make materials which don't show these kind of behavior. So if you want to make materials, you need to understand why this is happening. So to answer this why, we need to develop robust mathematical models. We need to understand those fundamental processes which are behind these observations, right? So my area of research is in that regime only. Means we try to understand the very fundamental processes which bring about such observations, and then develop mathematical models which can explain those observations. This is what we do. So before starting my work, let me also say how thermodynamics change in irradiated material. Like, most of the phase diagrams that they're all equilibrium phase diagrams. Although if you want to understand material which is driven away from its equilibrium state, we don't have phase diagrams, right? So what happens when the material is driven away from its equilibrium state? So that material moves in the free energy landscape, free energy and state variable landscape. At the and free energy landscape consists of global and local minima. So when the material is driven away from its global minima, so it may take it may occupy local minima. Local minima means the minima is the state of lower free energy by making via some kind of phase transformation, like by making precipitates, by making some other phase, and so on. So when we drive material away from its equilibrium situation, either by irradiation or by applying stress or under some pressure or anything else, so how the material responds. So the way the material responds will help us to understand how the material will behave in such non-equilibrium conditions. So in the particular case which we study, which I am studying, like the response of material under irradiation. So the material may respond by microstructure evolution, in which you might have nucleation and growth of new microstructures. You may have evolution of existing microstructure like grain growth and so on. Because of irradiation, you may have phase changes, like some phase was there, and that after irradiation, that phase evolved in some way, like formation of new phases, dissolution of existing phases, order disorder transformation. You may also have amorphization and so on. Or you can have just compositional changes. Compositional changes may include like radiation-induced segregation, compositional instabilities like which may be like spinodal type instabilities, or novel compositional changes which may not lead to phase separation and so on. Or you can have just the the smallest thing like species redistribution, but it doesn't lead to any any big observation like any change in the formation of a new phase and so on. Just species redistribution. That depends on the level of radiation the material is having. So I will discuss the mathematical. I will take one particular case and discuss how you can build a model to understand the observations, and then how after building a model, how exactly you solve it by developing algorithms and results, and some light I will put on what exactly I'm doing right now. So I consider I'm just giving an example of a binary alloy case. And so binary alloy like AB kind of system, which under irradiation can have several species. Right? Considering that A and B atoms are of similar size, so you can have A and B atom, you have vacancies, and you can have several kinds of interstitials. So I consider in my case, because the species are A and B species are of similar size, so they will make split. They will have dumbbell type interstitials. So you can have pure interstitials. You can have mixed interstitials. So if you want to understand the fate of the material under irradiation, we need to understand, you need to track all these species. What is happening with what? All right. And so you have defects like interstitials, you have vacancies. So there is a possibility of reactions of the defects with the vacancies. Even defects can also react with the atoms. So in fact, there are three different fundamental processes that we need to track. One is the defect production, how the displacement cascade, how the fundamental interaction of the radiation or the fission fragments with the material lead to the displacement cascade or lead to the production of residual amount of defects. And then how these defects diffuse. These defects diffuse via atoms, atoms on the lattice, and then how these species react, right? So defect production will put inside material the defects, which will lead to the atomic transport inside the material. So any concentration of any species, whether A, B, vacancy, the interstitial species, should depend on the diffusion process, the reaction, and the production of defects. So this is a reaction diffusion type equation that we have to construct. Now this is just the skeleton of the equation. We need to derive all these things, how the flux of each species will look like, when these defects will react with vacancy, like recombination reactions, or we can have a dumbbell change in the dumbbell type, like pure dumbbells, pure interstitials may convert to mixed interstitials and so on. And then at the end you have the source term, which drives the evolution of the concentrations. So I will not go into detail, but just I'll just tell you the how did I make the defect production model. So by looking at the structure of this cascade, like at the center you have vacancies surrounded by the defect and interstitials. So accordingly, at the center, to represent the vacancies, I use a dumbbell. I use a Gaussian profile. This is written by this is what I use with a certain FWHM, and then the interstitials were represented by a ring profile. Alright, this is a ring profile. So
Dr.
this
Dubey,
is, yes.
excuse me. Dr. Dubey, please conclude it, because 2 more lectures are in the queue.
Is it 20 minutes?
Yeah.
Fast? Oh.
20-minute time. Sorry.
All right. So I'll quickly. So I will not. Okay. So to mimic what exactly happens inside the material, I modeled this interaction as a random process. Random process, I mean, like suppose you are driving in a car and suddenly the raindrops start falling on the windshield. So the same thing happens inside the nuclear reactor, and each interaction with the material will dump some kind of a certain proportion, certain concentration of certain concentration of defects. All right, so this is what, like, I'm showing here. I'm showing the time series of all the cascades. All the cascades are falling on the material, and in each cascade we have a certain random concentration of defects inside the material. So this is the model which we get, which consists of 6 stochastic partial differential equations, and then for studying different different phenomena, you need to again derive the boundary conditions. So I studied 2 phenomena. One is the just to understand the composition fluctuations inside the material, and the other is segregation near the free surfaces. So for deriving to understand the segregation process at the free surfaces, you have to derive the reaction boundary conditions, which is this, like the flux of defects coming to the surface, and it will react with the surface, means it will either it will add material to the surface, or it will deplete material from the surface. So after you are done, then you can solve, and then you can exactly study the phenomena. So I use copper gold alloy for testing my model. Copper gold alloy has all those parameters which I needed, and to solve those system of equations, I use Gear method. Gear method is Professor Bill Gear from University of Illinois Urbana-Champaign. So I took his algorithm and then wrote the code and so on. I will not go into that. I'll just so these are the simulations. I don't have time to show you the simulations, but this is the observations, the composition fluctuation that I get. I took the Fourier transform, which is using FFT2W package, and this is what I found. I found that the patterns of around 19 nanometers stabilize in such a material. Now, depending on the irradiation condition, I can make fluctuations of smaller size or bigger size. All right. Similarly, for the segregation case, like I again use the same model with different boundary conditions to study segregation. I will run these simulations. These are interesting simulations. So at the left and right, we have free surfaces in both the cases. In one case, copper is a faster diffusing species. It goes and sits at the surface near the free surface, and gold is and gold comes inside. So you have depletion in the gold, and enrichment in the copper. This red region shows the enrichment. Copper is coming to the surface, and you see there is some movement in the surface also, because as you add up material at the surface, there will be some movement. All right. So yeah, so I'm also working on some new topics after I came to India. So those 2 topics are mainly metal-induced crystallization on under irradiation, and then one more thing which is very new nowadays, if nickel monosilicide synthesis. which has got very good applications in electronics, in microelectronics. Okay, so metal-induced crystallization means like we have suppose amorphous, say amorphous semiconductor materials like germanium and silicon. You can make crystalline germanium or crystalline silicon under irradiation and followed by annealing. If you irradiate that along with some metal like aluminum, silver, and gold. So I will not again go to detail because the time. And the one thing which is I'm working is a very new, which is under this work is under review with thin solid films. So synthesis of nickel silicide. Nickel and silicon has a very complicated phase diagram, but nickel monosilicide is very, very conducting. When you reduce the length scale or the size of nickel silicide form nickel monosilicide, which is very conducting. When you reduce its size even beyond 100 nanometer also, then also its conductivity increases. That is a good. So it can be a suitable replacement for cobalt silicide, which are used in nanoelectronic applications. So yeah, we already have got pretty good results, and this is under review. So that's it. So and then we are also working on some new modeling paradigms, like in Kahn-Hilliard framework, which is much more complex models. But this is under construction. I'm working on this development of the free energy of a material under irradiation. Anyways, so thank you. This is my conclusion, and I mostly do modeling of the material to understand a particular phenomena. And lately, I'm working on some more topics like metal-induced crystallization and formation of nickel silicide. And I'm also working on the models of those two materials. Thank you very much, sir.
Thank you, Dr. Dubey, for your nice and informative lecture on fundamentals, processes behind robust models, thermodynamics of irradiated materials, responses, dynamics of defects, and future aspects. Thank you very much, sir, for your nice lecture and for your valuable time to share with me. Thank you, sir. Thank you very much, sir. Thank you. The next speaker is Dr. Y. Dwivedi, NIT, Kurukshetra, Haryana. The topic of the lecture is barium tetraborate microfibers. Spermilities embedded in borate glass ceramics. Thank you, sir.
Thank you very much, sir. Professor Ajay Singh, thank you very much for inviting for the talk. It's indeed a matter of great pleasure to be part of this event. First of all, special thanks to Honorable Dean and the convener of this conference, Professor S.N. Tiwari, Professor Santuni Rastogi, Professor Lalni Azad, and other learned faculty members of the physics department for organizing this event and inviting me to be part of this. Since I can see Abmanu is in the queue, so I should be a bit quick. I will share just two major results. Hopefully this will be interesting for the audience of this conference. Let me share the screen and then we'll show you. If you can see the screen now. My talk is about barium tetraborate crystals that I grow in borate glass. It has two forms actually, microfibers and spherulites. I use one of the lanthanide ions, samarium, as a probe to see what there is optical effect, what the optical effect of these two structures, microfibers and spherulites. As sir already said that I'm working in physics department NIT Kurukshetra. This is my email ID. If there is any question remain unanswered, so please you can share with me on that. So because time is very less, I will be a bit fast to introduce lanthanide ions. Just a couple of slides. Lanthanide ions, atomic number 57 to 71 are considered as a lanthanide ions, but the extended family of lanthanide ions includes scandium, yttrium, and thorium as well, because some of the chemical and physical properties are a bit similar. The beauty of this lanthanide ions is that there is a partially filled or unfilled 4f ions are there. 4f electrons is there. And this 4f electron is shielded by S and P levels. That gives you very good advantage, optical advantages. Although name is rare earth, but this rare earth is actually not rare on Earth, because you see the maximum quantity that's reported in case of cerium is around 66 part per million in the Earth crust, while the least one, thorium, is even larger in quantity than the silver and cadmium. Lanthanide spectroscopy is unique in the way that the optical active electron is in the 4f configuration, and since the transition between this 4f is forbidden, though it partially releases due to that mixing of opposite parities, 4f 5d with 4f, and due to this some strength, some optical transition gets some strength, and that gives you a very unique feature like large radiative lifetime, and the energy levels are comparatively sharper because this optical active electron is not taking part of bond making with the host. So there are lots of advantages are there of these lanthanide elements that use in chemical catalytics, metallurgy, magnets, and phosphor purification of petroleum, batteries. Lots of advantages are there. There are particularly lanthanide group phosphors have advantages in case of X-ray storage phosphors, solar cells for the random lasing purpose, security, different probes in the case in field of biomedical, bioimaging, others. There are lots of advantages there. I will not go in detail of that. Time is less. Maybe some other time I can discuss. Today I'm going to talk about some glass ceramics. So this glass ceramics is a two-phase system. One, the base is the glass, and very few microcrystallites or nanocrystallites are there. So there is it's advantageous that this host have glassy nature as well as crystalline nature both together. So you have advantage of both. That can be proved with the help of electron microscope, that when you focus here, so you can see this disc like structure, and when you focus on this black spot, so which is the microcrystallite, so you can see the crystalline dotted feature. So in this case particularly, I have chosen this bored glass because it has a very high phonon vibration, around 1300 centimeter inverse, which is actually good for the down conversion analysis, and further it has a very good mechanical and thermal properties. People have already analyzed this nonlinear optical properties as well. So in that way, bored host is important. Let me start this microfiber synthesis. For the synthesis, the simple sample is very simple. That we have S3BO3 as a network former, and barium fluoride as a network modifier. So this we have tested several times, so nothing worried about that. This always form a very good glass material. And I use samarium ion as a probe ion to see any change in microstructures. So different concentration you can use, starting from 0.1 to more than like 1.5 mole %. Remember one thing, that since ionic radius of these lanthanides are very big, so it has a tendency to cluster, to form aggregate or clusters. So there is a very quickly these ions are show the concentration quenching, or you say emission quenching, with a little bit higher concentration. However, this concentration is not fixed for every element, and host to host it also vary. So to make this microfibers, I use two-step annealing method. First, to make glass, I heat it at 1000 degrees Celsius, so it melt, and then later on it cool down in stainless steel cast, and then we have a very good optical transmittance transmitting glass. That glass we kept for heating purpose, two-step heating. First at 410 degrees Celsius, that is already optimized much before, near the nucleation temperature, and then heat it at 540 degrees Celsius, which is near crystallization temperature, though the crystallization temperature is around 600 degrees Celsius approx. So we heat it for different hours as well, for example 10, 15, 24, 30 hours, even less. We start actually from 5 hours and something like that, but we optimize that if we heat it before beyond 15 hours, then in that case glass becomes translucent, and at 30 hours it's completely opaque, and due to the formation of big size crystal in that. So when you keep heating rate approximately 15 degrees Celsius per minute, in that case crystallization actually start near the surface. So there is a surface crystallization, or the bulk crystallization is there. So but in this case we keep this crystallization, this heating rate around 15 degrees Celsius per minute. So you can see that due to the some heterogeneity is present on near the surface and in the sample as well. So crystallization start from the surface and it branching inside the system. So what happened that this unique structure form, that upper layer is crystallized, that you can see the pink scatter light, and then here you can see the in between there is a transparent material. So it has some advantages. So when you heat it for 15 hours, then you can see around 0.2 mm thick layer surface is there. But if you heat it for more than 24 hours, then its thickness is around 0.5 mm. This but from the upper view, with the help of crushed powder, if you see under this electron microscope, you can see these fibers. We have estimated around 50 micron length at least. I mean, it is more than that actually, but 15 micron is unbroken, you can say. So and the diameter is approx 500 to 600 nanometer. However, in some cases, even it is more. We have heated it even at less temperature, and we see these fibers. But for 15-hour heating, you can see this type of fiber bundles are there on the surface of the glass slide. So for the analysis, actually, we cut this glass into 3 portions and take just clip off this one by polishing, very careful polishing, and then separate out the center portion as well. So and then we analyze this surface analysis and the optical analysis as well. For the X-ray diffraction analysis, this is the azimuth glass, the characteristic feature of boarded glass you can see here. When you heat it for 15 hours, you can see some spikes. Those spikes represent the formation of crystals in the lattice. If you heat it for 30 hours, so you can see the lots of crystals are there, but now these crystals are not only barium tetraborate, that means B4O7, but there are some other impurities, some other crystals also grow there. So approximately 30 to 35% of volume got crystallized. We also conduct this, calculate this absorption cross section, and this has lots of these peaks. You can see around 17 peaks are there. We can identify and easily assign that. But you see, in case of glass ceramics, this absorption cross section is a little bit higher than the glassy phase. This is a black line. But you see, this increment is not uniform. In some cases, it is too much, and some cases it is very small increment. You can see this is actually happening because some transition to transition. The nature of this transition, if it is a hypersensitive one, you can see good enhancement. Otherwise, there is a very less enhancement. So to explore much this absorption spectrum, we conduct this Judefold theory, and this Judefold theory, a very old one, I think more than 50 years. And that theory actually used for quantitative characterization of 4F transition. This described the 4F state, the perturbation of the 4F states by opposite parity, and the basis of 3 parameters. Since this admixing of 4F family level with 4F levels is a function of local crystalline field and the symmetry and the bonding nature, the local microenvironment near this dope ion. So that's why these 3 parameters, omega 2, omega 4, and omega 6, this actually represent the local interaction of lanthanide with the host. So in case of glass and in case of ceramic, glass ceramic, we find the same trend, like omega 2 is more than omega 4 and omega 6. So what is the significance of this? Omega 2 value is slightly higher in case of glass ceramics, and this is the reason that omega 2 actually represent the asymmetry and the covalence of lanthanide ion at the site. In case there is a crystallization is there, but even though it looks like that samarium ions don't actually diffuse in a crystal in a larger quantity, most of samarium ions are near the surface, so that they are actually in a very asymmetric area, asymmetric region. So we have also estimated some radiative parameters, and particularly if you see in this case of glass and glass ceramics, one transition 6H9/2, which is a very hypersensitive transition. So that transition value actually increased from 21 to 20.1.6. So this slight increment, even it is very important. And on the basis of this radiative probability, we have estimated the lifetime, and you see in case of glass ceramic, the lifetime is reduces 1.85 in comparison to glass, which is 2.02 millisecond. So the transition probability actually lifetime actually reduces. That shows the transition probability is more. So just I want to share this one, this photoluminescence spectrum of samarium dope glass ceramic and glass. Actually, we monitored this PL at least 5-7 different excitation wavelengths. But just I want to share one so that how it looks like it can be visible. So you see lots of transitions are there, but in case if you compare the glass and glass ceramic, so you can see the stark splitting is there, and the intensity is very high in case of glass ceramic. So direct evidence that microfibers are actually suitable for this to increase the emission intensity of samarium ion. Here you can see this is the visible image. I prefer to show it because it looks like Lord Ganesha. So this is very beautiful pink orange color. You can see yellow orange color. We observed the concentration quenching around 1.25 mole%. Actually, glasses and glass ceramics are good for this purpose, to have more and more lanthanide concentration, so that emission intensity you can increase and absorption you can increase further. So on the basis of this, so here one thing I missed that. So we have one this transition at 544 nanometer, and this one is 561 nanometer, and this 561 is considered as a hypersensitive transition, while this 544 is a magnetic dipole transition. Since this is it has a very small effect of microenvironment on this transition, so you can consider this as a reference, the reference peak to evaluate the change in microenvironment. So if you have a ratio of these two, so you will get some idea about how the microenvironment around lanthanide ion is changing. So in case of glass ceramic, we find it is around 2.9 in comparison to glass, which is around 45% enhancement. So that is actually matches with the what result we concluded in case of default theory. We have also calculated some parameters, important parameters, stimulated emission cross section, gain bandwidth, and optical gain. This is important for the laser characteristic. If there is a possibility to have a lasing action, you need to have this type of parameters. So in case of glass to glass ceramic, if you compare, it's every case it increases, gain is also increases. So glass ceramic is in that sense a very good one. So we analyze this decay curve also with respect to the heating hours, and you see after this 15 hour heating, this decay curves is no more mono exponential, and there is a two lifetime start appearing. Further, we also analyze this with respect to the ionic concentration, and you see till 1.25 mole per samarium concentration. Beyond that, it is a bi-exponential fitting is there, and there's a fast term is also appearing. So this fast term is appearing, as I said before, that lanthanide ions ionic radius is a bit big. That's why it has a tendency to. So ionic solubility is a limited one, and that's why it has a tendency to form cluster or in the form of aggregation. So due to that, it open up some pathways to relax the energy through the unexcited ions. That means excited ions to unexcited ions, and then different transition move to different transitions. That's why new terms are appearing. So the cross relaxation, that means the energy transfer between the identical ions, we try to estimate it, and then from the very lower concentration 0.25 mole %, we see that from 38 per second to it increases to 685 per second, and this is due to the several transitions have energy matching condition. So let's go for the other part. Just 3-4 slides, and then I will stop myself. So as for lights are there. So if you heat this borate glass with a very high heating rate, so some phase separation is appearing, and that phase separation is actually or the heterogeneity is lead the growth of grains and the branching in the crystals, and mostly it looks like this type that you can see in the image. The same thing mostly is it occur in case of polymer. It is reported silicon glass. It is reported. And also borate we have reported, but for this we have selected a different structure, different glass composition. One again we have S3BO3, and then barium fluoride, and then lithium fluoride as well. So here we have chosen two ions, samarium and YB, to as a probe ion. Maybe all result I'm not going to discuss here, but just one result I will show, which is really important. So to make this is for lights, I heat it at 620 degrees Celsius, and the heating rate is around 10 degrees Celsius per minute. So, but the time hours is also changed because now this temperature is close to crystallization temperature. So the maximum result, optimized result is at is for 10-hour heating. So this is the transmission electron microscope. You can see dark and bright field images, and the black spots represent the glass ceramic crystal formation here. So this type of structure, the cone, little bit cone shape, cone type of structure, two-head cone type of structures. You can see while you heat it for 10 hours. If you heat it for larger time, so you can see the big crystals like this, and this is the confocal images. This is for 12-hour heating. We also see that how the polarized light is actually affect this. If you can make some calculation for crystal growth in glass. So here in this case, you can see this is the simple glass particles. Well, under polarized light, you can see some color, blue, red, and yellow color as well. That shows the stress inside the crystal and formation of this micro nanocrystallines in the system. So we also, as I said, I clip off this upper surface and the middle portion, transparent portion as well, and then monitor the absorption spectrum. So again, the sharper peaks, like in case of microfibers, you can see this broad peaks is actually belongs to YB ions.
Dr. Dwivedi, please conclude it.
1 minute and I will finish. This is maybe just to give me 1 minute. I'm really sorry for that if I'm taking more time. So this is a PL spectrum. Just for comparison purpose, you can see here this is the black is represent the glass emission spectrum, and this sharper lines. This is really surprising, and this is the advantage of glass SEM. You can see this is the bandwidth is around 2 to 3 nanometer only. So it maybe act as a biolabeling purpose, and this is the confocal microscope image at 405 nanometer. You can see the very bright spot somewhere. You can see this is actually samarium emission. Further, we have then also see, you can see here this band have some dips are there. So these dips are due to the reabsorption of that emission by the samarium inactive samarium ions. When we compare this red one, this is the excitation spectrum, then you can see it matches well, right? So we also monitor the decay curve, like in case of microfibers, the optimized concentration of was 1.25, but here in this case, this is only 1 mole%, and the lifetime was in that case around 1.22 millisecond, but here in this case, this is 0.54 millisecond only. So this is a very important result that at a different micro environment, we have different that concentration and lifetime value. Thank you very much, sir.
Thank you, Dr. Dwivedi, for your informative lecture regarding the lanthanide spectroscopy, applications of lanthanide, doped phosphors, glass ceramics, its application, borate, microfibers, synthesis, and analysis. Okay. Thank you, sir, for your nice lecture. And I invite Dr. Ambrish Kumar Srivastava for the important announcement before the next lecture. Dr. Ambrish Kumar Srivastava, please.
Thank you, sir. So with the kind permission of chairman of this particular session, I would like to tell you that as we all know, the poster presentation is an integral part of any conference. So we are also featuring poster presentation in this international conference on futuristic materials. We have several posters presented in this conference. So I would like to request to you all to visit that posters and comment. In order to visit posters, you need to go to conference website, which is this. I will put these links on the chat box later on. So you will click on poster presentation. Is my screen visible to all? Okay. So you will see here all poster presentee listed by name. You will click on any name. It will lead to external link that will show the poster of that presenter like this. Now, in order to put any question or query, you have to enter your comment here and just you can also click notify me if you want to get notified whether presenter had responded to your query or not. So I would request again to all to kindly actively participate in this poster session as well. Thank you. Thank you all.
Thank you, Dr. Ambrish. Thank you very much. And I invite the next speaker, Dr. Abhimanyu Kumar Singh, Syama Prasad Mukherjee Government Degree College, Prayagraj. And the title of the lecture is Production of Gold Chitosan Core Cell Nano Structures Using Laser Ablation Techniques for Biosensor and Drug Delivery Application. So, Dr. Abhimanyu Kumar Singh, lecture. Please.
Thank you, sir. I am audible.
You are audible. You are audible.
Thank you very much, sir, for inviting me. I am also thankful to Professor S.N. Tiwari, sir, and Dr. Ambrish Srivastava for giving me opportunity to deliver invited talk in international conference on futuristic material. The title of my sorry. The title of my talk is the production of a chitosan core cell nanostructure using laser ablation technique for biosensor and drug delivery application. I organize my talk in the following outlines. First, introduction, in which I will discuss basic of laser ablation. Second, in experimental, I will discuss the formation of ultra pure colloidal gold nanoparticle. Then I will discuss the application of gold nanoparticle in biosensor and drug delivery application. Professor Shiv Bhardwaj in his plenary lecture already described the physics and chemistry of the biosensor and drug delivery application in details. So then my task has been easy. So firstly, I will introduce in nanoscience and nanotechnology are highly interdisciplinary, involving physics, chemistry, biology, material science, and full range of engineering discipline. I am not going to in detail. First, I will describe there are two ways of fabrication of nanomaterial. First, bottom-up approach, and second, top-down approach. Bottom-up approach is generally chemical method in which nanomaterial are synthesized by assembling of atoms, molecule, or clusters. Top-down method is generally a physical method in which the evolution of the property of a sample as its size is whittled down from the macroscopic towards nanometric length. I have used in this talk only laser ablation technique because it is very easy and rapid than the other techniques. This provide green and eco-friendly.
Excuse me, Dr. Abhimanyu.
Yes.
Please try to conclude within 10 minutes as we are already running out of time. So we request you to.
Okay, I will try to conclude. So the resizing and reshaping of the.
You have 10 minutes.
Colloidal nanoparticles synthesized by this technique or other technique are also possible by this technique. There are large number of parameters for controlling the structure, shape, size, and morphology of the produced nanomaterial. This is the schematic diagram of production of nanomaterial using laser ablation technique. When high intensity pulse laser beam is focused onto the target material, and explosive removal of matter from the surface of the target is observed. This phenomenon is known as laser ablation. In the case of liquid ambience, such a process leads to release a nanocluster to the liquid environment. Finally, we get the colloidal nanoparticle. The products are usually in the form of nanoparticle that remains suspended within the liquid medium and can be isolated by filtration and evaporation of a liquid. And this is the basic principle of laser ablation. Laser ablation means using laser light energy to remove a portion of a sample by melting, fusion, sublimation, ionization, and explosion. And this laser ablation takes the following process. When the high power laser beam is focused onto the target material, at first bond breaking and plasma shielding takes place during the laser pulse. And when the laser pulse terminates, plasma expansion and cooling process occurs. When the temperature of the plasma below the boiling point of the target material, then the formation of nanoparticle is formed. And this is the, here we can see that there is a rich color changes when the size is reduced. And this rich color size, the changes of rich color due to surface plasmon resonance, and the nanoparticle of silver gold is satisfy the visible wavelength. So this is the origin of intense color. And this is the experimental setup for production of gold nanoparticle, silver nanoparticle, and this is the photograph of the bulk and gold nanoparticle. In this I will describe the synthesis of a chitosan capped gold nanoparticle for application in biosensor. So this is the chemical formula of chitosan. And first, we prepare the colloidal matrix of the chitosan, and then we apply the for biosensor and drug delivery application. This is the nanoparticle produced in the water, and this is the cold nanoparticle produced in chitosan matrix. And this is the absorption of gold nanoparticle, bare gold nanoparticle. This is the capped chitosan gold nanoparticle. And from this, we see that the surface plasmon resonance peak is reduced when the capping occurs. And this is the fluorescence study of the colloidal gold nanoparticle and also chitosan capped gold nanoparticle. This is the emission spectrum excited by 342 nanometer, and this is the 367 nanometer bare gold nanoparticle. And this is the emission spectrum of chitosan capped gold nanoparticle. It enhances very much when the capping of the chitosan. And this surface plasmon resonance and fluorescence property of a gold nanoparticle make them potential lever for biological motivated experiment. And this can be used as a drug delivery application. In drug delivery application, I will drug deliver in the diabetic rat, or generally the current treatment option include the regular injection of insulin, which is very painful and inconvenient. Oral administration of the insulin remains a significant challenge because the insulin is susceptible to hydrolysis and digested by acid and enzymes in the gastrointestinal tract. So then for using gold nanoparticle, we deliver the insulin biorally. And we have used chitosan or amino-based acid, aspartic acid. In this experiment, I am using the aspartic acid. This is the formulation of capping of gold nanoparticle, and this is diagram shows the insulin loaded uncapped gold nanoparticle and capped gold nanoparticle. And this is the same diagram of the bare gold nanoparticle and aspartic acid capped gold nanoparticle, and this is the aspartic acid insulin loaded aspartic acid gold nanoparticle. And this is tested on the diabetic rat. And here is the last result. When the insulin bare gold nanoparticle delivered to the diabetic rat, there is no change. When the insulin loaded gold nanoparticle is delivered to the diabetic rat, there is a significantly change and almost 23% reduction in the blood glucose level to the normal level. And when the insulin loaded aspartic acid gold nanoparticle is delivered orally to the diabetic rat, then it is almost 75% reduction in blood glucose level. Thus, we can conclude that the synthesis of the ultra pure nanoparticle. This is the last slide. Using laser ablation technique could serve as an excellent carrier for the fast release of the insulin, the treatment of the diabetic mellitus. So therefore, capped gold nanoparticle loaded with insulin proved to be promising in controlling the diabetic. This is the important result and indicate that the transmucosal delivery of insulin by gold nanoparticle could be an excited alternative to painful and traumatic subcutaneous delivery. This is the acknowledgement, and thank you very much for.
Thank you, Dr. Singh, for an informative talk regarding laser ablation techniques, fabrication of ultra-pure gold nanoparticles for an application in biosensors and drug delivery. Thank you very much for your nice lecture and giving time in my seminar. Thank you very much, sir.
Thank you, sir.
Thank you.
Yes. I would also like to thank the chairman, Professor Ajay Singh, for coming to this session and participating in it. And I'm sure I wish to thank all the speakers, Professor Bhardwaj, Yash Dwivedi, Dr. Abhimanyu, and Santosh. It was a pleasure to hear all of you, and I'm sure we will have more such focused workshops, probably in near future when we can have offline contacts. Thank you very much. Thank you, Professor.
Thank you very much. Thank you.
So the next session of this conference will start from 2 p.m. and onwards. So I want to welcome you all to join that session also. Till then, I end this meeting. Thank you.